Good evening and welcome to the American Chemical Society ChemCom webinar series. Tonight we're going to look at Unit 7, which is Food, Matter, and Energy for Life. My name is Michael Murray and I work with the American Chemical Society here in Washington, D.C. And tonight I have with me two wonderful, experienced master teachers in Patricia Diver, who is in Wisconsin, and Barbara Sitzman, who comes to us from California. They will be walking through the sections with you tonight, and they'll be giving you some great insights into Unit 7. So again, welcome, and if you have any questions throughout the presentation, please do not hesitate to put them in the chat box or into the question box, and we will share them and everybody will be able to answer all the questions. So we will go ahead and get started. So Unit 7, again, is Food, Matter, and Energy for Life. Unit 7 is of course, one of the last 3 units in the textbook. Unit 5, 6, and 7 are meant to be standalone units after you've completed Units 1 through 4. And so some teachers teach Unit 7 right after Unit 4. Some teachers teach parts of Unit 5, parts of Unit 6, parts of Unit 7. Unit 7 has a lot of health and biological aspects of chemistry. So many health teachers and many biology teachers use this unit as well in their classrooms. So it can be used throughout and be used to help in many different courses. So the unit is separated into 4 sections, as all of our units are. Section A is talking about food as energy, looking at the energetics behind food. And then in Section B, we dive into carbohydrates and fats and look at their structures and how do they metabolize in the body and how do they actually work throughout the body. Section C is on proteins and goes through the structure and the makeup of protein as well as enzymes and how they're metabolized. And then Section D, we look at vitamins, minerals, and additives. The unit starts off with talking about the Parent-Teacher Association, Parent-Teacher-Student Association at the fictional Riverwood High School, and they're trying to change the vending machines in the current school to be able to best match the food required for a teenager. So the students are going to be presented with this dilemma at the beginning of the unit, and they're going to work through the unit, and they're going to come up with what are the best types of foods for teenagers to eat to give them the type of energy they need for their growing and their ever-active lifestyles. So that is the premise for this unit. Students are going to start the unit by actually completing a food diary. We have some sample food diaries for you in our teachers' resource materials, but students can take a 3-day food inventory and they can look and see what they're eating, and then they're going to actually use that inventory throughout the entire unit and once they get into, they're going to look at the calories and the energy in Section A. In Section B, they're going to analyze their carbohydrates and their fat intake. In Section C, they're going to look at their protein intake, and they're going to look at finally they're going to look at the vitamins, minerals, and additives in Section D. So they're going to actually use that food inventory and all of that data throughout the unit. And it's private for students, so you don't want necessarily students to share, but they can see what they're doing, and then they can get trends from others. And you also have the sample one so that students are actually saying exactly what they're eating, because that may be things that students may not enjoy sharing. So I'm going to pass the webinar along to Barbara, who's going to walk you through Section A, and then Pat will do Section B and C, and then Barbara will finish with Section D, and we'll all chime in if we have some suggestions throughout. But I'm going to turn it over to Barbara. Again, Barbara is in California. She teaches at the largest charter public school in the country, And Barbara's been using the textbook for many, many, many years and has been one of our teacher leaders for several years and is just amazing. So I'm extremely happy to turn it over to Barbara right now.
Thank you, Michael. And also my school just won the National Academic Decathlon, so it's really terrific. When I looked at the teacher's guide, they suggest about 34 days for this unit. We just finished our California State assessments, and my students just finished their water unit town meeting. So when I looked at the schedule, there were 28 days, but I can squeeze this into 28 days because the kids have a lot of background knowledge, and I'll kind of point that out as we go along. That's going to help them with understanding the material at a slightly pushed pace. They won't feel like it's pushed because we'll kind of light pedal some of the things. I find that this unit particularly will really help prep the kids for AP Environmental Science or AP Biology or IB Chemistry, all of these courses that we offer at our school that have quite a bit of organic chem in them. So the goals, we'll take a look at the goals, and these are beginning to focus, as Michael said earlier, on the information that kids need to know to be able to develop a proposal for this vending machine. So in the first goal, in the first bullet, they're looking at measuring energy. Now they did a lot of that in the second unit, but they were looking at calculating joules, and here we're just going to it'll be pretty easy for them to use the same kinds of procedures but calculate calories, the kinds of things that they've seen on the food labels. And then they are going to, in the second bullet, they're going to trace the flow of energy in food back to its sources and forward to its uses and dispositions. In other words, they're going to be connecting between the food that we intake in our body and how the body uses it. I kind of like to use the analogy that I used in the third unit when they were talking about petroleum, where people have to make some decisions, and there's a lot of risk-benefit analysis involved in how to use our limited supplies of petroleum. Shall we use it to tool around in our cars by burning it? Or is it important to also consider that we need to use it to make medicines, and we kind of like all the plastic goods that come from medicine? So, but in this unit, as you see in the third bullet, it's the body that makes the decision. You put the food in, but then the body kind of decides, is it going to use it to increase body mass, or is it going to use it to build more structures and so forth in terms of health? Okay, Michael, you can go to the next one. Thank you. All right, this is the first concept check in this section, and with the other concepts checks, the first question is just finding out background information. How much do the students remember about energy production from chemical reactions, and how to measure it and how to calculate it? And then the second one is kind of a bridge. We're moving, as I said a moment ago, from joules that we calculated in the third unit to calories. And then the third one is the looking forward, where they're looking at how this is involved in a healthful diet. And most of my students have taken some biology, so that helps them try to make that bridge and look forward to what this may mean. Okay, next. The USDA has the pyramid, that is shows the relative quantities of what should be consumed in each food group. And this photo has a little caption on it that suggests that kids take a look at that narrow yellow band. That tiny little bit represents all the amount, the limit you should have in terms of fats and sugars and salt. And they're going to be kind of looking at their diets to see how wide this yellow band is. Okay. In A2, students are making decisions, and I usually do these activities as groups. And following, I keep their group papers because this is some important information that they're going to need as they do that, the final proposal for the vending machines that Michael talked about. And he also mentioned that you need to be kind of careful here. They're keeping a 3-day diary of the food that they eat. And their diet, for some of them, may be determined by socioeconomic status. We have a lot of students who are on the free lunch programs, or cultural values. So this is something that should be independent. They'll do some sharing, but it's not going to be, you're not going to share with the class their actual diets. I find that the kids need a little help with the organization. I ask them, first of all, they've got a little bit of an indication in the picture here, but how they're going to organize this and set it up. If you feel more comfortable with having it in data table, the teacher resource materials have a data table for you that you can simply use for this. Okay. All right, in A3, we have the first investigating matter. And what they're basically going to do is take a piece of snack food. My kids are doing this on Thursday, and I've suggested that they look at the labels and they try to find something that has a lot of fat in it. They remember from burning biodiesel, they remember from burning candles and different fuels that it's important to have something that'll burn well. So they're going to bring in their own snack food to test, and they are asked to design their own lab. But there are some hints and suggestions that they go back to the C unit, 3C I should say, and look at these other labs, because this shows them how to set up their laboratory setup and how to construct the data table to record all the measurements that they need. Okay. Here we have a second concept check, and the first one is just asking them and actually giving them a page now about how to measure the energy from a combustion reaction. And their combustion reaction is they're going to set it up very much like the candle, but they're going to have a little setup with so that they can set their piece of food on and burn it, but the measurements that they take will be the same. And then they're asked to think about, and this is kind of the bridge, the connecting, they're going to think about where this energy comes from, and hopefully they're going to remember that that energy is stored in the chemical bonds of molecules. And then down in the third one, some of them have probably had biology, and hopefully you'll get someone to talk about cellular respiration. But in case they don't, this is coming up in the next section. Here's one of these beautiful potential energy diagrams where the kids are showing exactly what's happening. I kind of cushion this a little bit that it doesn't happen exactly like this, but this is a very nice way to represent it. And again, a place to reinforce one of the major misconceptions that kids have. Breaking bonds requires energy. You've got to pull them apart. And then the forming new bonds, forming the sugar and the oxygen, this releases energy. So it's a good place to point this out again. Over on the right, we've got a caterpillar, and we're looking at all of the energy that the caterpillar uses. And I asked my kids, have you ever seen anything like this before? In 3D, they looked at a car and they found out that the amount of energy was minuscule in terms of the overall energy that was actually used to propel that car forward. So not very much gasoline went into that. Most of it went into the other things. And the same sort of thing, the same kind of energy trace is valid for the caterpillar. Now we come to a chem quandary. And they're actually asked what they were looking at just before. You know, where does this mass of a plant come from? What is it? It grows into a big tree. Where does it get all of this mass? And sometimes you have to refer the kids back to photosynthesis and ask them to take a look at it. Most students haven't considered that this carbon dioxide produces the mass. Okay, next slide. Now we're looking at energy release and storage. So we've got a couple of pictures in this section. They're going to compare reactions of photosynthesis with cellular respiration, and I usually ask them to look at the two equations and chemical equations and tell me what they see that's similar and different. In other words, I want them to recognize, and they usually come up with this, that respiration is just the reverse process of the photosynthesis. So we have a picture here of things growing, and then we've got the energy being stored, and then we've got down at the bottom the bicycle. Then I also asked them to look at the ATP and ADP, and they probably studied this in biology, and they'll notice right away, because it's given to them in sort of a pinkish red there, that there's the tri means that there are 3 phosphate bonds and the di is 2. But again, you want to stress to them that the making new ADP bonds is what releases energy. There's more energy in the ATP than there is in the ADP, and this is what gives the energy to be able to ride that bike. Okay, go to the next one. There are lots and lots of tables in this section, so it's a good place for kids to get lots of practice in reading tables. In many of these tables that students will soon recognize depend upon the individual lifestyle, people's size, their weight, whether they're male, female. And so they're given the table, and then they're given some sample problems, and then a list of problems that they will solve. They're always amazed that sleeping actually uses a little bit of energy, not enough to really take care of all the calories that you take in with a bowl of ice cream, but it does use a little bit of energy. And so you can ask them about why they think that the body just makes that decision to use some energy while they're sleeping and breathing, and their heart is beating, and so forth. I always work through the example problems with students, and then this is something that is fairly easy. It's set up nicely with dimensional analysis, and so this is almost in my class, it's often a homework assignment then to go ahead and work the rest of the problems. Okay. All right, the A7, the energy in and out, refers to the ice cream sundae, banana split, whatever it is that somebody is eating, and then the exercises. And they're also given a list of some of the benefits of exercising, besides just losing weight and using up that ice cream sundae. And then they're given again a sample problem, and they use that table that we saw just a few minutes ago to look at those different activities to see how much bicycling would be required to use up calories and how much running is required to use up 150 calories. And then they noticed that running burns a lot more energy than the bicycling does. And as they go on, one of the really special questions in this section is they're given the idea that if you drink ice water, your body has to bring that up to room temperature, and that requires some energy. So, voila, maybe we can just drink lots of ice water and we can eat all the sundaes and french fries and whatever we want. So they go through and they do the calculation, and they find that if you want to use up all the calories in one serving of french fries, you're going to have to drink 26 glasses of ice water. They're kind of amazed at that. Okay. In the next section is making decisions. At the end of each section, if you're familiar with the other units, it's the same way. There is making decisions at the end of each section that really focuses on what the final project is, the vending machine project. So again, I have my students do this exercise. They're using the table that we saw a couple of slides ago to calculate the energy in various activities that they are involved in. And then they're finally asked what items are necessary in the vending machine, what would be best as they analyze their food diary. So I save these group papers again, and then just keep them in a folder for the class, and then bring them all out as they're working on their final project. Okay, next one. All right, here's your Section A summary, and as in the other units, these 3 bullets are the same goals that we looked at earlier at the beginning of the unit. They're boxed, and they're followed by a series of questions which refer to that section of the information that they're given. So it makes it quite easy for you to assign questions depending upon how far they've gone. I usually, in a section like this, I would assign 2 sets of problems. You could assign 3, some problems after each one, but I want to make certain that I'm checking for their understanding along the way in the middle of the section as well as near the end. Okay. And this final, this is in each section summary set of questions. There is one box that's quite distinctive. It has the green margin, as you see, and this is what I use as a prompt for the student reflection. My students keep composition books and in those composition books they keep class notes, reading notes from material that they read, questions that they have, section summary questions that they've answered, and most of the developing skills of little activities that they do as homework. And finally, at the end of the section, before they take a quiz on the section, I ask them to write a reflection. And this gives them my kids have trouble just write a reflection. They don't know what to do, but this gives them a nice outline of what should be in that reflection, as well as other things they think about. And even in the second paragraph, it shows them vocabulary words that it's probably a good idea to make certain that they understand these and use these words in sentences in their formal reflection. Okay, I think that does it for Section A, but perhaps, Pat, did you want to add some things?
I think it's good. Does anyone have any questions for us? Well, I guess if not, then I will move on to Section B, entitled Carbohydrates and Fats. In this section, students are going to discover the composition and functions of carbohydrates and fats. They will learn the role of these two molecules of life and the role they play in providing energy for life. They're going to review the structural similarities as well as the energy potential in each of these two molecules of life. Some time will be spent looking at saturated versus unsaturated fats. And in this section, students will also review the concepts of limiting reactants in chemical equations under various conditions. Next slide. Here we have concept check number 3. All 3 questions here draw on the student's experience from biology, so we're going to assume that most students that are taking ChemCom have biology in their background, where they have studied cellular respiration, they have some understanding of exothermic reactions and the structural differences between saturated and unsaturated fats. Section B1, carbohydrates are the products of photosynthesis and are energy storage compounds. They can exist in a linear chain, or that chain can link and form a ring. I get the students to look at the diagram and then try to distinguish or see the distinction between the starch and the cellulose molecules. At first glance, I find that most students are not able to distinguish between starch and cellulose. They look alike. So here it is important for students to notice the small differences between the two molecules. In digestible starch, both the glucose rings are attached to the connecting oxygen so that the oxygen is below the ring. We call that an alpha linkage. In cellulose, the oxygen is above one ring and below another. We call that a beta linkage. And in humans, we're able to produce the enzyme that will, through a hydrolysis reaction, break the alpha linkage, but we are not able to cleave the beta linkage. Next slide. When we take a look at fats, we'll notice that regardless of the fat, there is the COOH or carboxyl group. Students explore the distinction between the meanings of fat in common language as well as that used by scientists. How are these two molecules of life alike, and how are they different? In this section, they will also explore 3 types of fats: saturated, unsaturated, and the triglyceride. Next slide. While all fat molecules contain that glycerol backbone, fatty acids may differ significantly among fat molecules. Can be of different lengths and have a variety of degrees of saturation. So this schematic is showing the percent of saturated fat, the percent of polyunsaturated fat, and monounsaturated fat, which will lead us to the developing skills. Here, the students are looking specifically at butter and margarine, and they should conclude after doing the calculations that nearly all the calories in butter and margarine are from fat. B5 talks about hydrogenation, and here the students are going to, at least in my class, I have them discover what the hydrogenation process is, why is full hydrogenation rarely completed, and if it is, then the product becomes very brittle and hard and not really very palatable. But partial hydrogenation will cause some of the kinks in the unsaturated chains to be lost, and then the molecules can be packed closer together. So this is a way of taking a vegetable fat and converting it into a more solid product. And then you want to also make sure that you review with the students the cis-tran isomerism. This leads us then to yet another discovering or developing skills. And hopefully from this activity, students will learn that life is about choice. They will be comparing the fat content of ice cream to the fat content of yogurt, which leads into the ChemQuandary, which is having students focus on the idea, can a food ever truly be fat-free, and how misleading that might be. That actually, if the body doesn't use up all of the energy in that fat-free product, that the carbohydrates and proteins that make up the fat-free product may in fact be stored as fat in the body. We move on then to concept check number 4. Questions 1 and 2 are formative assessments just to make sure that students have an understanding of the concepts that have been delivered heretofore. Question 3 is connecting the students back to concepts that they were introduced to in Unit 3 and then which were expanded in Unit 4, which is offering us yet another example of how the chemistry in the community curriculum spirals to help facilitate long-term concept understanding. Section B7 is introducing limiting reactants, and here they use a macroscopic real-life example, the cake recipe, as an analogy to begin to have students understand about limiting reactants. Earlier in the course, they used the, I believe, the hot dog packages and the packages of hot dog buns. So they're coming back around to review limiting reactants. Modeling matter, B8, you might want to introduce this using the demonstration that's outlined in the teacher notes on page 717. This relatively simple demonstration provides a chemical example of the limiting reactant concept and involves observable chemical changes. The modeling matter activity leads the students through a practice in predicting quantities of product produced based on limiting reactants. And it starts off with macroscopic examples and then leads them into the molecular world of cellular respiration and the importance of having enough oxygen present because, as you know, oxygen is that ultimate electron acceptor, and without it, things kind of clog up at the end of the mitochondria. Section B9 and B10, here students extend the idea of limiting reactants to the quantitative analysis of chemical reactions. It's important before students begin working on the problems here to remind them that each substance will react in terms of moles first. So you want to calculate how much each substance will react in terms of moles and then convert the moles to grams. B11, again, is going to lead the students to the PIAT, the putting it all together at the end regarding the vending machine. Students are going to return to the 3-day diary or diet that they had from section A, where they analyze the energy content, and now they're going to analyze those same items in terms of identifying the source of that energy. Section B summary, again, here are restated in the orange boxes the review of the 5 goals that were set forth at the beginning of the section. And I usually assigned 1 or 2 questions from each of those sections, the ones that I felt were going to pull the greatest knowledge through. This query, again, allows students to connect the concepts that they have just learned to nutrition and how they sustain life in living organisms. And as Barbara mentioned, it really provides a prompt for a reflection that's going to draw all of the vocabulary and the concepts that they have just studied in this section into some well-formed thoughts for them. Between sections sections B and C is a highlighted feature of chemistry in the community, and this is chemistry at work. I think sometimes teachers have a tendency to skip over this section, but part of our role as educators is to increase student awareness of career opportunities. We never know when just one little chemistry at work or some other reference to a career opportunity is going to ignite an interest, and a student then has a new career path to follow. Are there any questions on section B?
I'll look in the question box and see, Pat, so we'll just pause for just a moment for any questions.
I do not see any. Is
Me
there
neither.
something?
We can continue.
Okay. All right. Section C talks about proteins. Proteins happens to be one of my most favorite topics to talk about, so I'll try to contain myself. The essential question here, and there you go, why are proteins essential to living organisms? This section focuses on proteins as builder molecules, as well as enzymes that control rates of chemical reactions, which is essential to life. Oftentimes here, I will burn a cube of sugar and show that you can't, our bodies just can't ignite a cube of sugar, or it would destroy all of our cells. So the rate of a chemical reaction then becomes essential to life. Students in this section will also learn about the building blocks of proteins, the amino acids, and how the chemical properties of their unique R groups allow the protein to fold into a functioning macromolecule within a living organism. They also learn how amino acids form peptide bonds and how the linkages of the amino acids determine their function. Students in this section will also learn that some amino acids can be synthesized by humans and how other amino acids need to be part of the diet. Concept check number 5. Question 1 immediately reinforces the structure of carbohydrates and fats and why that structure determines properties that are so different. Question 2 takes the students back to the collision theory that was introduced in Unit 4 to further illustrate what has to happen in order for a chemical reaction to occur. Then question 3, as is the norm in all of the concept checks, leads the student into the next subject or concept in which they are going to engage. Section C1 talks about proteins, their function, and their structure. It's important to review or have the students rediscover that proteins are important in living organisms in that they serve in structural capacities. When we look at ourselves in the mirror, we are looking at structural proteins. Proteins are also important in regulation as in hormones as well as enzymes. In chemistry, we call them catalysts. In biology, we refer to a catalyst as an enzyme. Transport. In order to transport the oxygen through the bloodstream, a very important protein, hemoglobin, is involved in transporting that oxygen, releasing it just outside the mitochondria, where it will then become the ultimate electron acceptor and form some water down in there. So I often tell students that they have to breathe oxygen in order to pee water. Proteins also provide protection, as in antibodies found in our immune systems. It's important that students review the structure of amino acids, that they understand that all amino acids have a central or alpha carbon, that connected to that alpha carbon is an amine group, a carboxyl group, a hydrogen, and then what makes the amino acid unique, the R group. The amino group of one amino acid is going to react through a dehydration synthesis reaction with the carboxyl group of a neighboring amino acid to form a peptide bond. In my class, I have necklaces that are alanine and glycine, and in one hand they hold the OH for the carboxyl, in the other hand they hold the hydrogen for the amino group, and then they stand in a row and the enzyme comes along and causes the water to form, and when the water drops to the floor, a peptide bond is held between the students, so we end up with a great big long polypeptide chain around the room, which then leads us into the discussion about the primary protein, the idea that a linear amino acid chain is that polymer that emerges from the messenger RNA as it is translated, and then that primary protein will begin to bend and fold depending on the chemical properties of the R group attached to the alpha carbon. So this leads us into a discussion about hydrophobic and how the hydrophobic side chains will tend to fold inward to be protected from the watery environment of the cell, whereas the hydrophilic side chains will extend out into the watery cytoplasm of the cell. You can also look and see that some of the amino acid side chains are acidic and others are basic, and they will be attracted, those opposite charges will attract. We also take a look and discover the cysteine, where it's located, and the idea that it has a little bit different structure in that it has a sulfur in there, and that disulfide bridges will form between those cysteines, and then holding that whole protein molecule together to keep that true functional form are weak hydrogen bonds, which may be expressed in the beta sheets or in the alpha helixes. So I have gone on more than probably it talks about in that section. The next section is modeling matter. Examine and practice writing structural formulas for amino acids and polypeptides. From 20 different amino acids, students soon realize that there is an unlimited number of possible proteins that a living organism can synthesize. Sometimes you might, if you have molecular model kits, you might have students build some simple amino acids to help in their understanding. A few years ago I discovered some flat molecular puzzle pieces that are produced, and I'm not promoting a company, but they're produced by a company named CellZone. And if you are teaching dehydration synthesis and hydrolysis reactions, sometimes these flat puzzle pieces will enable greater understanding when students just really don't have a handle on all of the bonding angles that the 3D molecular models, sometimes that just gets in the way. It's also important to note in this section that, not discussed in ChemCom, but I think it's important that with all the bioinformatics that has caused computer notations of amino acids, that there has been a move away from the 3-letter abbreviations assigned to the amino acids, which is very cumbersome in the computer notations, and that each amino acid has been assigned a single letter. Section C3 talks about protein in the diet. This section moves students from the molecular world of proteins to the macro world of foods which contain protein. Here students are reminded of the digestive process, which breaks apart complex proteins that are consumed and then reassembles them into functional proteins for the overall health and function of the individual. Further, there are some amino acids that the body cannot manufacture, and they have to be consumed in the diet. So that can be a point of discussion as well. In this section as well, the students are introduced to the dietary reference intakes, which will be valuable in the next activity. So C4 is going to get the students to begin to think about needed protein intake in regard to metabolic function at various stages in one's life. Why does a pregnant female need more protein intake than a geriatric male? That leads us to concept check number 6. And here question 1 again reviews that importance of the amino acid structure to how that protein is going to fold. Question 2 is reinforcing the need to constantly be taking amino acids in. And then question 3 is going to again lead the students, which we're going to discuss enzymes. And finally, we get to an investigating matter. It's been quite a while since we had one. In this investigating matter, we're going to illustrate the rate of decomposition using different biologicals that contain an enzyme. And this exercise goes a little bit further in that it also throws another variable in that they're not just using fresh samples that contain the enzyme, they're also using samples that have been boiled. And in my class, sometimes we have done this particular exercise, but sometimes we go back to an old biology lab that I did. and that's on the Smiths and the Jones. One won a case of fresh pineapple. The other family won a case of canned pineapple, so they decide to throw a block party. And the students have to predict, will the Jell-O blocks made with the fresh pineapple be more palatable than those made with the canned pineapple? And the enzyme bromelain prevents the gelatin from setting. So most students will say, oh, the fresh Jell-O, but then when they come back the next day and the control is set and the canned pineapple is set and the fresh pineapple is mush, it is a very similar illustration as to what is being discussed in this investigating matter.
I could just add one thing, Pat, to this.
Oh, absolutely.
In terms of the substances that you're testing, you do test liver. Kids get all grossed out about that. And I have found that if you take frozen chicken livers, it's much easier to cut little pieces and have it all ready for the kids when it's frozen. If you let it defrost, it's really a mess. And the other thing that works best is you need a fairly new bottle of hydrogen peroxide. You want to store it in the refrigerator, but then take it out and have it at room temperature when they use it. Of course, it will decompose much more rapidly.
Okay, thanks, Barbara. That's very true. I always used calf's liver because I use that in some of my biology classes, and and I've had good results. But certainly, chicken livers are a little bit more palatable to slice and dice. So investigating matter on enzymes then leads us into a discussion in C6 about how enzymes work. And here students are going to review another biology concept, the lock and key model. I would also recommend, just the other day, Dr. Tim Herman was in my office, and he works at Milwaukee School of Engineering with me in the biomolecular design department, and he came in with some foam models that's kind of this lock and key, but in addition to that, the model actually allows for the induced fit, but then he has it arranged so that you can actually show the role of the inhibitors in the process, which leads to a really rich discussion on the various roles of pharmaceuticals. So again, linking some relevance to real-world experience and how drugs work when we're looking at how enzymes work is, I think, very powerful. But throughout the ChemCom book, as you know, there's a number of Scientific American conceptual illustrations, and this particular illustration is remarkably well done. That takes us then to the next Chem Quandary, which is this is really a fun activity. Students get tired of chewing and they know that they can't swallow that saltine cracker. It's important to note, however, in this activity that roughly 25% of the population doesn't produce enough salivary amylase to convert the starch completely to glucose. So about 25% of the students are not going to have that sense of sweetness. But I wouldn't say don't do this activity because of that, because there's going to be a number of students who are going to be able to relate that experience. So I find this to be a fun and real meaningful activity for students. That then leads us to, and as ChemCom always does, leads us to the next activity where they're going to do another investigating matter. In this activity, students are in the last activity, they learned that temperature does alter the integrity of a protein, making it ineffective by denaturing it. But here they're going to continue to investigate the role of temperature, but they're going to take it one step further and look at pH as well. That it's very important for the pH of the system to be constant in order for an enzyme to work. It's a 2-day lab. Day one, the students are going to prepare their samples, and then the second day, they're going to set up their hot water bath and do the Benedict's reagent test. Barbara, did you have anything to add to that lab?
I don't think so. Thank you.
It's a pretty straightforward lab, I think. Then moving again to the last activity of this section, again, it's leading the students one step further to that putting it all together activity at the end. Students are going to return to the 3-day diet from Section A, where they analyze the energy content, and in Section B, they identified the sources of that energy. And now in Section C, they're going to analyze their 3-day diet, their 3-day inventory for proteins. The section summary, again, in the little orange boxes, will review each of the goals that were established at the beginning of the section. And then we end up again with the reflective question. I'm going to go ahead one more, Michael. Why are protein molecules essential to living organisms? And again here, vocabulary and allowing students to reflect on all of the learning that has just taken place. Any questions on Section C? Then I think I'll turn it over to you, Barbara, for Section D.
All right, thank you, Pat. In Section D, which is the last formal section before the final project, students will explore the chemistry of vitamins, minerals, and food additives. So if you see in the first bullet, the first goal is to take a look at the key vitamins and their importance to the body. And in the second one, they're going to be taking a little closer look at the vitamins, and we're going to see which ones are fat soluble and which ones are water soluble. Then in the third bullet, they're going to look at minerals and learn the difference between trace and macro minerals. Next bullet is about food additives, and this is where they start looking at risk-benefit analysis. And finally, they'll look at a couple of the techniques through labs that they'll do of titration, and where they learn to measure the quantities of additives, and then they'll look at chromatography and how they can identify what's in their foods. Okay, next we have a concept check. And again, the first two questions are pretty well going through and reviewing the sections in B and C. And actually the 2A is reviewing the enzyme. And then in 2B, we go to speculating what a coenzyme might be. And in 3, we're looking at the students now know about vitamins and minerals, and we're going to, and you might question them to see about, this one asks about the mass, and so you might ask them about where these vitamins come from, and some of them will probably talk about the tablets, vitamin pills, and so forth, and that kind of gives them an idea about the comparable mass of proteins, fats, and carbohydrates that's needed in the diet as compared to vitamins and minerals. Okay, on the next slide, we're looking at another one of these tables. And this table is based on gender and age. And also, students will notice right away, probably, as Pat mentioned earlier, the needs of pregnant and nursing mothers is quite a bit different from the other females. And then as we go through and we take a look, we've got developing skills where they're going to use this information. And again, this is a pretty straightforward exercise that I usually use as homework. Give them some practice using the table and applying this to their own diets. Next we have an investigating matter, and a very good pre-lab is really essential for this. One of the things I do is they're doing vitamin C analysis, so one of the things I begin with is just demoing the reaction between the iodine and starch. I usually use the starch pellets that come in packing materials. These make kind of a nice way to get the starch rather than trying to cook up cornstarch in water, which is kind of a mess. You want to emphasize that the starch must be added to each reaction. If you've done a demo, they see that that's necessary. And one of the mistakes I find that kids forget to put the starch in, and it just turns kind of brown. There are some things that I might mention about the materials for this lab. First of all, that I use the starch pellets, and I just give each one of the student groups one of the packing pellets and tell them to just mix that in a small beaker with a little bit of distilled water. It doesn't dissolve completely, but that really doesn't matter. They just need a little bit of it in there to work. And you notice in the picture that the well plate on top of a piece of white paper gives a nice contrast so that they can really see that reaction just as soon as it appears. In terms of vitamin C, you can use a stock bottle, but I simply use a vitamin C tablet. I usually have some of them at home. And I can take either a 100 milligram vitamin C tablet and 100 milliliters of water, or I can use 1,000 milligram vitamin C in 1,000 milliliters or a liter. And so then I get, as you see with the sample problem, which we'll go through in a moment, if you look at the third step, what you want to get is a solution that's 1 milligram of vitamin C to 1 milligram of 1 milligram to 1 milliliter. And so either the 100 milligrams in 100 milliliters or the 1,000 milligrams in a liter will give you this standard. And I wouldn't worry too much about whether you have that exact, because the whole purpose of the lab is for them to compare different beverages to find out which ones have the most vitamin C. So if your standard's off a little, that's okay, because that's going to be the same for the entire class. And then they're going to titrate using Burrell pipettes. So they're going to do this by numbers of pipette drops. So let's take a careful look at the sample problem. First of all, the first thing they do is they set up a standard. So they're using 25 drops of the vitamin C, and then they need to titrate to find out how many drops there are in a milliliter. So they need a small graduated cylinder, a 10 milliliter one is good, and then just find out, using some water, how many drops from their Burrell pipette will be equal to a milliliter. In the sample case they've used here, it's 30 drops, and that kind of depends upon how narrow the orifice of the pipette is. And then they will multiply it by that standard, the 1 milligram of vitamin C per 1 milliliter of vitamin C. And then finally they check these 25 drops of their standard, and they count the number of drops. Not forgetting, they mustn't forget to put the starch in. They count the number of drops of iodine that's required for the blue-black color change to occur. And so once they have done that standard, then what they do is just simply use, instead of their standard, and kids, you need to kind of work with kids in this pre-lab so they understand they've done the standard now. And so now they're going to replace that 25 drops of vitamin C from their standard solution with 25 drops from their beverage. I asked the kids to bring beverages in, but once they've seen the demo, they'll recognize that blueberry juice isn't going to work very well because they won't be able to see the color change. So they can bring in anything that seems to, any kind of beverage that doesn't have a strong color to it. My kids are kind of surprised sometimes that some things that say there vitamin C is added really don't have very much vitamin C in them. And then they're absolutely amazed when they bring a fresh orange in, the amount of vitamin C there is in the orange in comparison to the others. So also one of the things that they want to look at with this is, what else could contribute to the amount of vitamin C in a beverage? And usually after a while they'll think about the age of the beverage, and they'll all run around and look at their beverage containers and see what the date is. It is it past the due date, or have they had it open for a while? Is this something they had for lunch and now they're using it, and maybe some of the vitamin C has broken down? Okay, so that's quite a bit on that lab. Pat, did you have anything else to add to that?
No, I like your idea about using a starch palette. That sure cuts down on the mess of making a starch solution.
Okay, I think we can go on to the concept check, Michael. Oh, did I miss something? Oh, I did. Excuse me. This is another table that they're working with, and this one has minerals in it, and they're divided into the macro minerals and the trace minerals. And of course, kids have a little misconception about this. They think that these are big minerals and little minerals. And so you need to really point out to them that we're looking at the same amounts, but that these macro minerals are ones that they may need more of, where the trace ones are the ones that can be limiting reagents. And Pat talked to you earlier about limiting reagents. It may just be a small amount that they need, but this is sufficient to counteract the deficiency conditions. And the kids are always quite interested in that third column where they're looking at the deficiencies. There is also a second table that's given that they'll be using in the developing skills that will come next, where they do take a look and they have to identify where they fall in the age group for males and females, and to see the amounts of these different minerals that they need. Okay, so we could look at the next slide. This, again, is a fairly straightforward calculations that they do. I do the sample in class. They've just had to look up the DRI on the for the 16-year-old female in the example on the table in the preceding page, and then they simply use a nice dimensional analysis to figure out how many cups of broccoli they need if they're going to get all of the iron that they need just from broccoli. Okay, and then we move on to the concept check, and here again the first one is the background check. It's really a nice place for you to review ionic and covalent bonds and have that, and that ties very nicely to reviewing that the vitamins are molecular, and some of them are polar molecules and some are non-polar, and that brings the water solubility that's important. And I usually talk about cooking and losing the water-soluble vitamins in the pan of water if you use a big pan of water to cook your vegetables in. And also the idea that you need to be kind of careful if these substances are not water soluble, because they're going to stay in your body for a long time. They're going to be absorbed by the fat, so you want to be careful of the amount that you use. And then the looking ahead is beginning to take a look at food additives and see whether they know of anything that is added to food products, because that's going to lead us to some risk-benefit analysis. So the next slide has a table again, and this table is on food additives, and it also on the left has one of these wonderful Scientific American illustrations where in the top bowl we've got a layer of water and with oil floating on top. Then in the next one, we crack an egg into it, and of course you got to stir it up a bit, and they can see how the emulsification of these substances works, where you've got the micelles forming, where you've got the egg yolk that is water soluble on one end and a non-polar or fat soluble on the other, so the fat globules all just collect around, form this little micelle in the solution, and the other, you can see some of the long arms going out from the egg yolk molecules that are attracted to the water, and this, with help of beating it up, will form mayonnaise, or whatever you're trying to form. But you can form form a solubility between by the just hooking these together. You can emulsify the oil and water mixture. The food additives then are all listed, and then now we're going to analyze in D7. We're going to analyze for food coloring additives and investigating matter. This one doesn't take all period. It's a fairly short one, but they need to leave their samples to dry. They're going to do a little bit of paper chromatography and also calculate the retention factor, which is calculated by the distance moved by the dye sample. As you see, they've measured to the center of the dye sample, and that's the numerator in the box. And then the length that the solvent goes is your denominator, so your RF value is a decimal. The picture shows the material that's being put onto the dye sample being on a watch glass. What we use for dye samples here, because we're looking at food additives, is we use M&Ms, Skittles, and food coloring. Now, I have the kids do this in a well plate, and I come by and I put a drop of one color, let's say red, dye in one well, and in the next well I give them a red M&Ms, and in the third well they get a red Skittles. And then they put just a couple of drops of water on the M&Ms and the Skittles, just a minimal amount, because you don't want to dilute the dye, and stir it up, and the dyes are soluble in the water, so they have all three. And the only problem with this is getting enough dye on the paper, so they need to draw a line with a pencil line and then put their dots, label them. You see that dye sample A is labeled in the picture. And I have them dotted and then wait a little while and come back and dot when it's dry and dot it again and dot it about 3 times. They want to get plenty of dye on there. And then they will do a dye sample B and C for the 3 different things. Now, I assign different groups have different colors. So somebody's doing yellow, somebody's doing red, and somebody's doing, usually we can find green in the samples. And then they can compare these. You want to collect class data and compare them. Now, if you want to leave them overnight to dry, be certain that they mark the top of the solvent front, because they may not be able to see it the next day, and also the dye may fade quite a bit. So it's a good idea for them to use a pencil to mark both of these. And one year I was able to get some chromatography jars, which are a little bit larger, and some wide chromatography paper, and that worked a lot better because they had plenty of space to put there to separate their 3 dyes. But it does work all right just with the narrow and the 100. You want to use a 100 milliliter graduated cylinder, so it's as wide as possible. And they've used a stopper up there to keep that just pretty well seal that chamber and keep the piece of chromatography and paper in place. I usually pull it up over the lip a little bit and then stick the stopper in. Pat, do you have anything to add to this?
The only thing that sometimes I have done in this particular lab is I will use coffee filters and cake pans and put the water in the cake pan and then just invert the coffee filter, and that allows the solvent to move up and then separate the colors.
Okay, thank you. All right, then in the next section, we're going to be taking a look at regulating additives. And you know, many of my students have heard that some salad bars note that they don't use nitrites, and because some people are allergic to them. So we're going to be looking at some of the federal regulations about various different additives. And these regulations, the URLs for them and so forth, are given in the teacher's edition in the margin. So you may want to give these to students to pick up some additional information. You see the reactions that occur as we have the acid, stomach acid, and the nitrite, and then how that all in the stomach again, the nitrous oxide, nitrogen, the nitrous acid reacts then with the protein during digestion, and it forms nitrosamines, which are carcinogenic. And so this is a place where they start looking at what are the kinds of things that we need to consider. They may know that some red dyes have been prohibited by our federal regulations, and so this is really is a very interesting section, and it's a place that you can expand upon. And on the next slide, they're given some of this information, and they're asked to consider, again, the risks and benefits. And the idea that if we don't have some sort of preservatives and so forth, then we'll end up perhaps with things like botulism, which are even worse. One of the things I do is I cut an apple, take a couple of apple slices at the beginning of this section in the book, and I dip one in water and I dip one in some sodium nitrate solution, and just set them out on the front counter, and the kids start kind of watching them, and they notice that the slice that has just been left in the air without being dipped in the nitrite turns brown pretty quickly, and the other one stays for quite a while. So they can kind of understand why this is something that is really attractive if you're setting up a salad bar and you don't want the salad ingredients to turn brown and look rather unappetizing. But it's important to look at both the risks and the benefits. Okay, next one. Here we look at artificial sweeteners, and one of the things that you might ask students to look at is compare the structure of sucralose and then have for them the structure of sucrose, and let them compare the two, and suggest to them that one of the things that chemists do when they're trying to find a substitute, an artificial sweetener, which will be good if you're on a diet, to a molecule that might possibly be used as a substitute, take a look at the structures. So if they take a look at the two, they'll see that there are a lot of OH groups on both of them, and that's one of the things that early chemists took a look at, was how are these structures similar? And when they find something that's kind of similar, that's a possibility for a substitute. And this brings us to food labels. And so they're going to be doing a food additive survey. They're going to bring some food labels from home. Oh, please tell them to, before they rip all the labels off the cans in the cupboard, to please put a little piece of tape on so that when other people come along, they'll have some idea of what's in that can. So they're going to need a little bit of help with this, and but they will start trying to analyze the food labels and look for the additives that are involved, and make a list of these additives in a data table that they set up. And then they're going to have, as you see in 3B, they're going to have 4 vertical columns, and where the food product was found, the purpose of the additive, if known. Now they had some of them given in the table, but they may need to go to some of the websites that are given to find out other information, chemical formula for it, and other information. So this really involves doing quite a bit of research on their part. And then taking a look to think about whether they think this additive is worth the possible cost of some kind of problems that it might cause. So this is really sending them toward trying to decide what kinds of foods to offer, what these foods might have in them to offer in this vending machine. So then in the final making decisions section, again, this is the final one they're doing just before the vending machine PIOT. The teacher is going to specify, you want to limit things a little bit, so the students just look at 2 vitamins and 2 minerals, or if you're short of time, like I'll be, I'll probably just have students look at 1 vitamin and 1 mineral that they want to analyze. And they're going to go back and look at their 3-day inventory and look for the presence of those vitamins and minerals, and take a look at how much is in their diets. And so this is going to be the place where they really start looking at what should be in that vending machine. Here we have the section summary. Again, this is mimics the goals, and there are some questions following each one of those, and you'll want to decide which ones you want your students to answer. And remember, the first two were about vitamins, and then we had minerals for the third one, and then we were looking at food additives, and then in the final bullet we're looking at lab techniques for separating and identifying the components of a solution. And here we have the Section D summary, which again I use as their reflection and their chance to demonstrate to me their understanding of the new vocabulary that was introduced. Okay, and following this we have the putting it all together. And when I do these final projects, I think it's important that students have individual responsibilities as well as the responsibility of the group of students to work together. And actually, I have formed my own groups all the way through until this point, and my kids were very excited when I finally granted their wish on Monday and let them choose their own groups of 3 for the final unit. And so group work is coming up with a policy statement. The teachers' resource materials give you a possible set of rubrics that you can use, and actually in the textbook the students are told what they need to do in terms of using an analysis of their 3-day food diary as the basic data for their recommendations, and they have to come up as a group to formulate recommendations. Then individually, each person is going to write a report that they're going to submit to the PTSA. So that's going to be, they're going to be individually graded on how they do that. And then the written report, the sequence is given in their textbook. And as I said earlier, they've got suggested rubrics for this. And I use the suggested rubrics as the basis, but then I kind of tailor them to fit my own students. So Pat, do you want to add anything to this?
I think you have covered it very well, Barbara.
And so are there any questions?
Yes, we'll definitely open it up for questions. Thank you, and I want to thank everybody. More information about ChemCom is at the websites shown here, and you can email us at chemcom@acs.org. I do want to definitely thank Barbara and Pat. Pat works in Milwaukee now. She was a high school teacher and has been a ChemCom teacher leader for many years, and thank you so much for your experience, Pat. Pat has a background in biology, so she definitely loves this chapter, as we can tell. So I want to thank both Barbara and Pat, and if you have any questions, we'll be happy to answer them. So I'm looking into the question screen right now. And while you're thinking of questions and maybe writing them down, I wanted to let you know that the last 2 webinars of this spring season will be, one will be next Tuesday, May 7th at 7 o'clock, and it'll be an intro to ChemCom. And we'll also have another intro to ChemCom on Tuesday, June 4th. I don't see any other questions, so I want to thank you again, Pat and Barbara, and thank you all again for attending. And have a wonderful evening.